Wow. None of them are scientifically perfect, so.
Do you have I do for endocrinology, and I feel like when you don't accept the images by yourself, it's like the recording that's like elaborating on the images right now. Like maybe you're listening to his lecture and you miss all the things, and you're like, oh, now I don't know what we're talking about, I just don't know what thing he said from the back. I think it's recording it. I felt that. Listening to that image section, like on the that image, like really understandable.
Yeah, I really like that. I love it. No, yeah.
I don't feel like you're going to be missing out.
Okay, let's start. Yeah.
Right off the bat, I was just wondering about the hormone. In the syllabus, it says that it's could be about from that 29 and in the parent about it, it says like
Yeah, I screwed that up. So look at the one that's on the in the actual, not on the paragraph. So that's last year's. I forgot to change that date.
which one's on?
So the one that's in the actual syllabus, on the daily thing, that's the time. So I think it's coming up in a like not to the end of next week.
Yeah.
So next Wednesday. So, and all I'm looking for that is just finding a partner and telling me what your hormone's going to be. So that's it. It's not more than that. All right. So as you might have guessed, the exams are not done yet. We hope to have them done by next Monday. No promises, but that's the target time. So you guys did a lot of writing. Okay, so today we're going to start off doing stuff with reproduction, and specifically mammalian reproduction at first. So it does beg the question, what is a mammal? So what do you guys think? How would you define a mammal? What makes a mammal different from a bird or from a reptile? Yeah? Lactate. Okay. That's mostly true. There are some birds that actually lactate. Your common pigeons out here produce this thing they call a crop milk. Comes out of their part of their throat called the crop, and they will actually produce a nutritive fluid, much like milk, and they will feed that to their chicks. So milk production or some sort of a nutritive fluid production you give to your offspring is not actually diagnostic for a mammal. Yeah? Mammary glands. Okay, so that's one. Definitely. There's nothing else in the animal kingdom that has mammary glands. What else? Yeah?
Hair growth?
Hair growth. Yes. What about whales? Dolphins? They're mammals. Do they have hair? The answer is yes, actually. That's very little amounts. So yeah, hair is another one. There's two other diagnostic features of mammals. Yeah?
Live birth?
Sorry?
They give birth live, they don't.
No, it's actually, lizards and a lot of snakes actually do live birth. And believe it or not, a lot of sharks give live birth. And there's a whole series of people that are studying reptile lizards actually have a basic placenta when they give live birth. So that's not diagnostic. Yeah?
Warm-blooded?
Oh, birds are warm-blooded. sweat glands. Sweat glands are the third. And you brought up live birth. Not all mammals even give live birth. So the marsupials have live birth, but it's the monotremes. The monotremes. No, there's two monotremes. Anybody know what they are? Yeah?
Platypus.
Platypus. So I got to show these things to you because I'm so proud of them. I did my sabbatical in Australia last year, and we were walking down a stream, and this is what we saw swimming around in the water. A platypus. And here's one that's a little bit higher magnification. So yeah, they are the coolest creatures. They're actually only about this big, so they're pretty small. But the cool thing about them is that, they have this, it's really this sort of leathery bill and this kind of weird tail. And the first time the Australians sent stuffed specimens back to England to the big experts in zoology at the time, they basically thought it was a hoax. They tried to see where the stitching marks were for taking the duck bill out and putting it on the mammalian body. It wasn't until the Australians actually sent a live specimen back that they realized that there was in fact a real creature. And the amazing thing about them is that they are also the only, as far as we know of, the only mammal that produces its own venom. And the males have a spur that they have on their elbows that they only produce the venom during the breeding season. And they seem to use it mostly to attack other males. But it's apparently, I was talking to some folks who were studying this down at one of the university I was at, and they were studying the venom for all its different purposes and things like that. But apparently the venom from a platypus is so potent that it will hurt excruciating pain for 3 or 4 weeks. It does not dissipate, and there is no known antidote that will decrease that pain. So that's where they're trying to figure out why and how it's working to see if they can get different kinds of anesthetics out of it. Anyhow, that's one of them. The other one, which is got to see even closer, is this creature, the echidna. So the echidna also is an egg-laying mammal. And they're quite a bit like porcupines. They have these big spikes on the back of their tails and on their sides. So live birth, not diagnostic. Milk, not diagnostic. Basically, you have 4 things to diagnose a mammal. Mammary glands, hair, sweat glands, and here's a good one for you. How do you, everybody looks, probably read stuff, they said, people have found this new fossil mammal. Mammary glands do not fossilize. Hair doesn't fossilize usually. Sweat glands certainly aren't going to fossilize. How in the world are these people telling that this is a mammal? Got a bunch of bones sitting in rock, and they say this is an ancient mammal. Does anybody know? Yeah, it's the other diagnostic thing about mammals. Bones of the inner ear. The 3 bones of the inner ear are unique to mammals. The stapes, the incus, and the anvil. Okay, so what we're going to be talking about for the next little bit, just sort of thinking about, just keeping that in mind, that these are the diagnostics of a mammal. A lot of the rest of this stuff is not unique to mammals. So we're going to start talking about mammalian reproduction. Next week, we'll talk about some of the more crazy things about endocrinology and reproductive endocrinology in non-mammals. Okay, so when we're starting to, and when we talk about mammalian reproduction, we've got to talk about it because there's similarities, but it's quite different in males and females. Okay, so today we're going to talk about males, mostly because males are simpler. Not simple, simpler. Okay, so if we're looking at the endocrine regulation, these are the main players. This is of the of reproduction. So one of them, the first one is GnRH. So this one's going to be stimulating FSH and LH release. LH, really important mostly for sperm production. We also have LH, which is primarily used for androgen production, and that androgen is mostly testosterone. Prolactin is not a particularly important hormone for males. Oxytocin is really important for ejaculation. So again, as we talked about before, it's a contractile hormone. So it's a contractile tissue. And then we have the androgens. These are the really important ones. We have primarily testosterone, but we also have, it's certainly in development, we're going to talk about this a little more next week, DHT, dihydrotestosterone. And there's lots of other androgens that are floating around, but these are the two primary ones. We also then have estrogens. Estrogens are amazingly important for mammalian male behavior. So most of the male behaviors are regulated by estrogen. And then progesterone, prolactin, the prostaglandins, all of the rest of these are really important in female reproduction, but not really in males. And then finally, the last one is inhibin. And so this is going to decrease FSH release. So these are the major players in mammalian reproduction. Now, if we look at testosterone production, testosterone production and LH release are going this sort of episodic or cyclical fashion. And there's a peak about every 6 hours of LH and then also testosterone production in the male. And this will then stimulate the production of all of the other things that the testosterone is doing. During puberty, this decreases so that we have it about every 3 hours. So that's primarily how we get the major increase in testosterone that regulates male puberty, is through that decrease in the cycle length. So decreasing it from 6 hours to every 3 hours. That's going to increase the total amount of testosterone we see. And so if you look over the lifespan of a human male and their testosterone levels, we see 3 major peaks of testosterone. This fetal peak right here, and the one at the neonatal period. So this is just after birth. These two are primarily to create the primary sexual characteristics. Okay? So one of the things that we have to do is we have to distinguish between primary and secondary sexual characteristics. Primary sexual characteristics are those parts of the anatomy that are involved in actual reproduction. In the case of males, sperm production. So the testis, the epididymis, the penis, all of these things are primary sexual reproductive organs. And those are all created early in development, either as a fetus or shortly after birth. We then go through this period, this pre-pubertal period, after this approximately 6 months to a year of the neonatal period, where in fact there's essentially no testosterone being produced by the male child at all. And then, right at puberty, we start to see this massive increase in testosterone. And this peak is when we start producing our secondary sexual characteristics. So for human males, that's decreasing the voice, that's facial hair growth, that's extra muscle production, all these things that help attract a mate, but aren't actually involved in sexual reproduction itself. Does that make sense to people? So it's attracting the mate rather than actually doing the mating. And those are all the secondary sexual characteristics. The peak, by the way, in a human male of their testosterone is about age 18 to 20. So, all you guys in the room today, enjoy it. It's all downhill from here. Okay? And so then it'll start to drop slowly until it's senescence, and that's the point where the males can no longer produce enough sperm to reproduce. There's been this debate over this years or centuries on whether or not there is such a thing as male menopause. It looks like there probably is a point at which most men fail to produce enough testosterone to generate enough sperm to actually fertilize eggs. But that's also, there's still debate on that. But it does decrease over the rest of the lifetime. Okay, so we take a look at the primary sexual characteristics, things we have to do. It's all taking place in the testis. So the testis has a number of features that we have to be aware of. So the testis is surrounded by a scrotum, and inside are these seminiferous tubules. And so these are the seminiferous tubules are densely packed coils of tubes in which the growing sperm are going to be sloughed off into. So you're going to have all of these sperm that are being produced, they're going to go into these seminiferous tubules. They then will be transported down the seminiferous tubules into this area, which is the ductus epididymis, or the epididymis. And they will stay in that epididymis for about 12 days. And then they will slowly, this is where they mature, and then they will slowly mature and then move into the ductus deferens, also known as the vas deferens. And this, the sperm will stay in here until they're ready to be ejaculated. And so if a human male, this whole process from the production of the beginning of the production of a sperm cell to the time that it's mature and ready is about 70 days. This, by the way, is why during men who have had vasectomies or have had birth control by cutting the tubes still have to wait for about 2 or 3 months before they engage in sex again. Otherwise, it's possible that the sperm that are being stored can still fertilize a woman's eggs. So a lot of times they'll talk about failure rates of contraceptives. And there's actually a reasonably high failure rate of sterilization, male sterilization. But it primarily occurs because they have sex too soon and these sperm are sitting there and still ready and matured. Okay? Okay, so this is sort of the gross anatomy of the testis. If we look at in more detail, this is what we see. Now, this is actually one of my favorite figures about male reproduction, but it takes a few minutes to understand what it is they're showing you in this picture. So, this is essentially, if you go from here to here, is essentially a time lapse of what's going on. So you start with the production of the spermatogonia. You go through mitosis to make this secondary spermatocytes. And we're going to talk about this whole process in a minute. But then it goes through to create the spermatids, and then you get the more mature spermatids, and finally the spermatozoa. So think about this as sort of a time lapse, going from the base here all the way to the end where you get the spermatozoa. And this will help think about how this figure is working. Okay, so one of the two most important types of cells in getting this whole process started are the interstitial or the Leydig cells. And these are going to produce our testosterone. The second one are these Sertoli cells. And the Sertoli cells have 6 major functions. The first function is that, and I'll put it over here, the first function is the creation of this basement membrane. And it is going to be separating all of these interstitial or Leydig cells and from the rest of the testis from both the Sertoli cell and all of these growing sperm. Okay? So the basement membrane is actually turns out to be incredibly important. So why would people think that a basement membrane is vital in the testis? It is forming essentially a blood-testis barrier. So that blood is not able to go past this basement membrane. So why create that barrier? Yeah?
Separating your immune system from accessing your sperm, so it doesn't treat it as like an other and prevents stroke.
Yeah, but so why would the body's immune system treat a sperm as other? Why is it technically is? Yeah? Because it's haploid. Not only is it haploid, but we have what else is happening before we even get to the haploid stage? Yeah?
Crossing over can create genetic anomalies that aren't necessarily foreign, but might not be recognized by somatic cells.
Exactly. That's the whole purpose of sex, right? Is to mix up the genes. And you don't just do that between the male and the female, but you also do it within each one of the sperm cell and each of the egg cells. And you're mixing up your genes. So the sperm are actually foreign to the body. And if the immune system had access inside the testis, then the immune system would attack all those growing sperm. So in fact, that is one of the ways in which people are exploring still for having male contraception, is to get some sort of a drug or a chemical that would get rid of that basement membrane and allow the man's own immune system to come in and essentially sterilize it. So people are still working on that. I don't think I think they can do it in mice already, but I don't know if they can do it in people yet, primarily because you also want it to be reversible. Okay? But this is one of the ways in which they do that, and that basement membrane is produced by the Sertoli cells. Okay, the second thing the Sertoli cells do is they create a peptide called androgen binding protein. So ABP, androgen binding protein. And that is secreted into the seminiferous tubule. So this is inside. And this is important to realize. This is the inside of the seminiferous tubule. So this boundary here is inside that tube. This is outside. So these are the capillaries that are bringing the blood. So if we come back to this picture, we have the ABP, the androgen binding protein, being produced here, and it is going into the seminiferous tubules and traveling down the seminiferous tubules into the epididymis. And it is taking then testosterone. So it's the androgen binding protein. It's binding the testosterone and transporting it from those Leydig cells, from this part of the testis, to the epididymis. And what it's doing there, what the testosterone doing, is creating the production of a nutritive fluid. And that is then bathing these growing sperm and allowing them to get the nutrients that they need to continue to grow and mature. Okay? So that's the testosterone coming down here. So, basement membrane, androgen binding protein. Another thing that they do is create the production of STP. So this would be the third thing they do. And STP stands for steroidogenic stimulating protein. And what it is doing is it is permissive for LH function. So it does that by increasing LH receptors on the Leydig cells. Okay, the fourth thing the Sertoli cells produce is a fluid. And this fluid is also released into the seminiferous tubules. And this is the fluid that then allows the sperm to move down to the epididymis. So they're producing this fluid. The fifth thing, the Sertoli cells stimulate the production of inhibin. And so just like we learned before, inhibin is going to inhibit, through negative feedback, the release of FSH. And then the sixth thing is phagocytosis. So the Sertoli cells, for any damaged sperm or these spermatozoa either that are not developing properly or developing too slowly, then the Sertoli cell will phagocytose those, break them down, and get rid of them. Okay, so 6 major functions of the Sertoli cell. One last thing to think about before we start talking about the endocrine regulation of this. And this is the production of sperm. So we have the spermatogonium. So these are the cells that in this time-lapse thing are right here, right next to the basement membrane. So these are the early cells that are going to end up becoming sperm. The first thing we need to do is we need to make lots of them. So there is a mitotic proliferation of these spermatogonium to create spermatogonia. So you get lots and lots of proliferation, mitotic proliferation. Once you have enough of these spermatogonia, then you can start building what's called a primary spermatocyte. And this is when we start the meiotic process. So we go to 4x, total the number of chromosomes. Then we go through meiosis 1. This will then drop us down to 2x. And then we go through, and they become what are called secondary spermatocytes. And then we go through meiosis 2, and now we're down to 1x. And these are now called the spermatids. And the spermatids then start to develop into spermatozoa. So here's a picture of the spermatid. So it's already gone through all of its meiotic divisions. It's now down to 1 copy of your chromosomes. And then in the process of creating the spermatozoa, we are now going to greatly compact the DNA inside the head of the nucleus. We start to produce the flagella, the flagellum, that's going to allow the sperm to swim, and we're going to create this thing called the acrosome. An acrosome is going to have all of the important enzymes that are going to break down the zona pellucida around the egg and allow the sperm to eject its DNA into the egg. Okay, so this is the maturation that takes place primarily right there at the Sertoli cells, but then fully matures down in the epididymis and the vas deferens. Yeah.
So just to clarify, is the primary spermatocytes, is that going through meiosis 1 or?
No, the primary spermatocyte is just before undergoing. So the primary spermatocyte is going to undergo meiosis 1. So this is mitosis all the way to here. And now, once we get to the primary spermatocyte, now we're going to go through meiosis.
So is that the same thing as spermatogonia in terms of its like composition?
No, because the spermatogonia is only going through mitosis. So in order to start meiosis 1, we're going to have to get double our chromosomes.
Oh, okay.
So it's that doubling of the chromosomes that is going to define the primary spermatocyte. Yeah?
Is the spermatogonium the same thing as the, it's just like the singular form of the next one's spermatogonia? The
Spermatogonia? Yeah,
just
multiple.
Like the multiple. Okay.
Yep. This is one spermatogonium. This is just multiples. So that's the whole idea there is to get an enormous amount of mitosis to produce the number of sperm that we need. Yeah.
Does meiosis begin in the deductus epidermis or the the tubules?
So here we go. Good. So all this is taking place here. So we're going through the spermatogonium. They're all going through mitosis here. So they're going through mitosis until they can get to the spermatocytes. And then we start undergoing our meiotic divisions to get to our spermatocytes. And so that's why I'm saying think about this as this time lapse. You start with the spermatogonium, you go through the mitosis to create lots and lots of more spermatogonia, and then you start going to the primary and secondary spermatocytes as you go through the meiosis to create these spermatids. And then what's called spermiation is you go from the spermatids and start creating the spermatozoa, which is the creation of the acrosome and the creation of the flagellum. Okay? So all of this is taking place over time.
In no particular place.
No, it's all right here. So this is before they get sloughed off into the epididymis, into the seminiferous tubule.
Okay.
So once you get to this point where they start to grow their tails in the acrosome, this is when they are sloughed into the epididymis, the seminiferous tubules, and they get moved down to the epididymis. Okay? Yep.
I'm wondering why in the image it says mitosis rather than meiosis.
It's both. So you got to have both of them here, mitosis and the meiosis. Because the mitosis is creating all the cells that are going to go through this process. Okay? Yeah?
So you mentioned that testosterone is needed for the fluid to provide nutrients?
Yep.
Okay.
All right. So this is your basic anatomy. Are we ready for the endocrine regulation? Yes.
Sorry, where is the nutritive fluid that testosterone is?
Well, we're going to get to so there's two things. So there's the fluid that's produced by the seminiferous tubule. It goes into the seminiferous tubules. Okay? And then we also have testosterone, where is it? Creating this nutrient fluid down here in the epididymis. Okay? So there's fluid being produced in both places. Okay? All right. So the first thing we think about is LH. So LH is coming up through the capillary and stimulating the production of testosterone from the Leydig cells. So that's its primary purpose, produce the testosterone. Okay, then we also have FSH. And FSH is also coming through the capillaries. This is because, remember, they're both being produced in the pituitary. So FSH is going to reach the testis and is going to have 6 major functions. The first is that it is going to stimulate the production of the spermatogonium. So if we come back to here, this mitotic division is all stimulated by LH. So that's where, sorry, FSH. FSH. Okay, so FSH is going to be stimulating this mitotic proliferation of these cells. It's also going to be stimulating the final movement from spermatogonia to the primary spermatocytes. Okay, so that's the FSH. The second thing that FSH does is going to stimulate the production of androgen binding protein. So it stimulates the Sertoli cells. We're going to increase the amount of ABP. The third thing it's going to do is it's going to stimulate the structural proteins that are important in the basement membrane. The fourth thing we're going to do is stimulate the production of STP. And then STP is going to come down to the Leydig cells and increase the number of LH receptors. The fifth thing is FSH stimulates inhibin. And then inhibin is going to go back into the capillaries to inhibit FSH release. And then finally, the sixth thing is that FSH is going to stimulate the production of more Sertoli cells. So Sertoli cell mitosis. Okay, so FSH, named follicle stimulating hormone, is primarily named because of its role in female reproduction in the stimulation of follicles. Okay? LH, luteinizing hormone, again named primarily for its role in females in the production of estrogen. But it's the same roles, so you can think about it exactly the same way in the male. FSH is primarily involved in sperm production. LH is primarily involved in steroid production. Okay, so the LH is going to primarily, in both males and females, be for the production of the steroids, and the FSH for the actual production of the reproductive cells. All right, and the last thing we have is we think of the testosterone. And testosterone has 5 major functions. The first is during development, we get the descent of the testis. So it comes out of the body, creates a scrotum to allow it to be outside the body. Does everybody know why it's important that the testes remain outside the body? Yeah?
Does that have to do with like the temperature?
Yeah, the temperature at which sperm is produced needs to be about 4 degrees Celsius lower than body temperature, 4 or 5 degrees. So that creates some really interesting issues. There's this fascinating story that I read about a number of years ago in which it was during World War I, And the U.S. Army took all of its soldiers and sent them over to Europe to fight in the trenches. And a bunch of them were sent during winter. And so they handed out wool underwear. And they fought in the trenches. And when a lot of these young men came back home, it turned out they couldn't get any of their wives or girlfriends pregnant. And the question that people in the military were asking was, why? So here are these young men, you know, the virility of the American male is all of a sudden we sent them to war and now they can't have kids. This was a major, major problem. And it turned out what was going on, they figured this out, was that the wool underwear was so good at retaining heat that they were essentially cooking the sperm. So the sperm were not able to get low enough temperature to be produced. So within actually how long did it take for this to be reversed? About 70 days. And then all of a sudden, everything went away. So interesting stories about, you know, why these things become necessary. However, there's a really cool story that just came out a couple of years ago. So there are mammals that do not have descended testicles. All the dolphins and the whales have their testes inside the body cavity. And so this is a 200 or 300 year old mystery that was just solved a couple of years ago, which was how do whales and dolphins, males, produce viable sperm? Anybody have an idea? Yeah?
Maybe using fluid inside, next to the testes.
Use fluid. Well, except that you've got to have this. We got to be regulating temperature. How do you regulate the temperature? Yeah.
Does the area where like the testes would be not have the same kind of fat around them like the rest of the body, so that it keeps it at a lower temperature than the actual body temperature?
Yeah. Yes and no. So no, it's actually inside the blubber.
Oh, it
It's
is.
that big blubber layer. You know, in a good whale, the blubber layer is about this thick. So that's what's insulating these whales. And the testis is actually well inside the blubber layer.
Is the blubber layer just cooler
Nope.
for whatever reason?
Yeah?
Do we know why sperm production needs like a lower temperature environment than normal mammals?
I don't think we do.
So it's not like a sperm mutation.
No. But one of the ideas for many, many years was there was a mutation in the what are called the cetaceans, which is the name for the group of whales and dolphins. And the cetaceans that allowed them to produce sperm at a higher temperature. But it turns out that's not what happens. And the way this was finally cracked was right here in Cape Cod. A small whale washed up dead. And they, about 6 months earlier, I don't remember where it is, but they had just brought online an MRI for big obese people. And so they took this small whale with the big MRI, and so before the thing could rot and decompose, they put this whale on a truck, drove it to the MRI, cleared everybody out, and stuck this whale in the MRI and did an MRI on it. Isn't that cool? Now what they discovered was that what the whales were doing was that they were sending the artery of blood down to the tail. And the tail has much, much thinner blubber. And it's right next to this cold water. And so when the blood finishes and comes back in the vein, it's much colder than the rest of the body. But then it goes back into the body and gets warmed back up. But before it gets back to the body and warms up, that vein goes right past the testis. And that's how they're able to cool their testes, using the blood that goes through their tail and back past the testis. Yeah?
So if well, probably die if you remove its tail, but if it were to live, would a male like dolphin be infertile?
If what? If you cut off its tail?
Well, like if let's say if it lived and it just didn't have that tail.
Yeah, an interesting question. Was that? There was a movie I showed our kids a couple of, was it? The dolphin that lost its tail and ended up with
The prosthetic.
a prosthetic tail, right? What was that movie? Dolphin Tail. That's it. Right. So yeah, there's actual real life. There was a dolphin that had lost its tail, and they produced a prosthetic for it, and learned how to swim around. That's an interesting question. I don't remember if that dolphin was male or female, but if it were male, you're correct. It probably would not be fertile because it wouldn't have that system. I hadn't actually thought about that. That's a cool idea. All right, exam question. Next year. What happens if you cut the tail off of a dolphin? Could it breed? Okay, so yeah, some neat stuff. And this is a major issue in human medicine also, because there is a syndrome. It's called cryptorchidism, or cryptorchidism. In which the testes do not descend just before birth. So crypt, you probably can't read this, can you? C-R-Y-P-T, crypt, and then O-R-C-H, orchidism. Where that word comes from, I have no clue. But the syndrome is that in males, and it's not uncommon. It's uncommon, but it's not rare, that a male will be born with undescended testicles. And now this is, easily taken care of. This male child is immediately just, as soon as they get born, immediately whipped into surgery, and they surgically have the testicles descend and bring them out. And so this is no longer a problem. And but traditionally, this was something that not often, like I said, it's very uncommon, but it's not rare that this would occur. And so all of those individuals would have been infertile because their testes were not able to regulate the temperature. Yep.
Are most males in general, do they have their testes internally before they descend?
Yeah, because during development, they're developing inside the body, and so there's actually this descent that takes place just prior to birth. And that's why one of the reasons why you get that big peak during the fetal period of testosterone. And then one of the things, as I said, that's for the primary sexual characteristics. And one of them is the descent of those testicles to allow that to occur. Yeah?
Yeah, I'm wondering why it might be advantageous to keep the testes from dropping during development. Why does this descent happen right after or prior to birth?
Well, it takes place. It's just part of the development.
Oh, it's just,
Right. So the testis ends up just being developed inside the body, and then it needs to descend. Yeah.
Is that also why when male babies are born, they have a tendency to have like a swollen scrotum because of the increased testosterone before they're born to bring down the testes?
That's an interesting idea. I don't know if that's the reason.
But I know that's usually what happens when they're born.
Yes, male babies generally have very outsized for their size testicles, which then shrink later. And it's got to be some fluid issue, and I just don't know what it is. Yeah.
If the drop occurs right before birth, is there a higher incidence of the syndrome in premature babies?
That's a good question. I don't know. My guess is no, but because I don't think it's not like a week or two before birth. It takes place earlier than that. But it is something that neonatologists have to deal with. Okay, so that's number one of the things that testosterone does. Number two is that it initiates the meiosis portion of the production of sperm. So we're going to come back to this figure. So we have FSH driving this, the mitotic divisions and the production of this primary spermatocyte. But the meiosis 1 and meiosis II are stimulated by our testosterone. The third thing is that testosterone feeds back primarily on LH to as part of the negative feedback. So this is the stuff that we've already learned. The fourth thing is that it goes to the androgen binding protein and then out into the seminiferous tubule. So this is where we're going here. So we're secreting that testosterone, and then it's getting the testosterone down there where you get this production of the nutritive fluid. So that's its fourth role.
Can you? So it's the testosterone is moving into the epididymis for the nutritive fluid. That's the fourth one?
Yep.
Okay.
So it's getting, and it can't get there on its own
I guess they're through.
because it's a steroid,
Yeah.
and the fluid in the seminiferous tubule is an aqueous fluid. So it's water. The steroids don't like to be in the water. So it has to have this binding protein, the androgen binding protein, to get it down through that fluid to the epididymis. And then the fifth aspect of this is that the testosterone affects a lot of these other cells using various paracrine regulators. And we're starting to learn a lot more about those, but I won't cover those in this class. But they are affecting a lot of the mitosis and everything else of these other Leydig cells and things like that. So it's all part of the paracrine regulation. Yeah.
Can you just repeat the third function again of testosterone?
Yeah, so that's the negative feedback.
Oh.
So it's going back up to the brain and to the pituitary. Yes?
So we should consider testosterone paracrine regulation of other cells like it's permissive ability? Or is it like not really necessary, just like adds to the other cells?
No, it's probably permissive for, yeah, because by doing all of those other things with those other cells, it's going to essentially have a permissive effect on production as well. Yeah.
Did you say that paracrine regulation was around Leydig cells around it, or the certain other cells?
All of them. Yeah. So the Leydig cells and the other kinds of cells that are there. These what are called myoid cells, but the fibroblasts, all of these things are being regulated. Yeah.
Do these hormones have trouble getting across the basement membrane? Or how do they?
So, well, the testosterone doesn't because it doesn't bind membranes at all. LH doesn't have to because it's just binding right here in the Leydig cells. The FSH, the receptors are across these, so that's how they're stimulating the Sertoli cells. Other questions? Okay, so that's the regulation. Let me show you just the process in a little different way, just so you get an idea here. Okay? So in this case, what we have, the pituitary. The pituitary is releasing FSH and LH. So the FSH binds to its receptor. It's a cyclic AMP receptor, stimulates the production of DNA, RNA, protein synthesis to get your androgen binding protein. And then the Leydig cells, the LH is going to stimulate the Leydig cells to produce testosterone. Testosterone is going to go bind to the binding protein, get out into the seminiferous tubules, and down to the epididymis. We also have testosterone doing a systemic roles. So in this case, it's going to create our secondary sexual characteristics, and it's also going to be converted into the brain, where it goes to estradiol, where we get behavior, male behavior. Okay? So this is just a different way of visualizing how this whole process is taking place. Yeah.
So when people say that, so usually like with aggression, people say, oh, like the testosterone influx, it's really estradiol the whole time.
Yes and no. So the way in which it works is that the androgen, the testosterone, gets to the brain, goes into the brain cells, and the enzyme, aromatase, is in the brain, in that cell, and it converts the androgen to estradiol. So if you injected a male with estrogen or estradiol, you will get the behaviors. You skip the process entirely. Yeah, it totally freaked everybody out when this was finally discovered. I was like, wait a second. The male hormone has to get transposed into the female hormone in order to get aggression. Then why is it that females don't have more aggression than males? Yeah. Yep.
Yeah, also going off of that, like, the amount of, like, testosterone that affects aggression is very context dependent. And so it's not always, like, high levels of testosterone equals aggression. So it's...
Correct. Well, it also depends on what kind of behaviors you're talking about, too. So one of the ways in which you can dramatically decrease aggression and reproductive behavior in things like horses and bulls, cows, and things, is to castrate them. Okay? So, castration, on the other hand, in human males, does not decrease aggression. And so it's almost like clockwork. Every 10 to 15 years, some conservative in the country decides that the way to deal with rapists is to castrate them. That's going to be the punishment, and it will decrease their behavior. And it doesn't work. Why? Because rape in humans is essentially not a reproductive behavior. It's an aggression, it's a power behavior. And testosterone, in conversion to estradiol, has no impact on aggression and aggressive behaviors. It really only decreases sexual behaviors. And it doesn't really do that in men as well either, unless it takes place generally before puberty, and then not even then. So they used to do this fairly routinely in Europe, castrate boys. Does anybody know why? Yeah?
So they can sing.
Because they can sing. They were called the castrati. And one of the famous things about castrati is that they were very famous for their sexual prowess. So I don't know how that was documented, but that's what everybody said. So apparently it doesn't really have that much of an impact in humans on sexual behavior either. Yeah.
I know that sometimes they say not to consume things that would have higher estrogen levels in men because it can cause them to have more female-like processes. How much estrogen does that end up changing those properties then? Because sometimes I know it can have organizational effects instead of just other effects.
So it does. And I don't know about from consuming food, but one of the things that's very common and well known among weightlifters who spend a lot of time taking anabolic steroids for building muscle is that one of the things they will develop are breast tissues. Why? Because all that testosterone gets converted into estradiol, and the estradiol then goes and stimulates breast production, just like it would in a female. So, yeah.
What's controlling where the estradiol is going, whether to the brain to affect behavior?
So to affect behavior, the estradiol isn't going anywhere. It's being produced in the exact same cell that it's affecting. Okay? So what you have is, this is a neuron. And so the testosterone comes into the neuron, gets converted through P450 aromatase into estradiol, and the estradiol affects that particular neuron. Okay? So it's not like, for behavior purposes, it's not like the brain is producing estradiol to go to the rest of the body. It's being produced right there. Okay? All right. So now let's try to integrate what we've known before with what we now know. Okay, so here's our hypothalamus. Hypothalamus is producing GnRH. The GnRH is producing LH, which goes to the Leydig cells to produce testosterone. And testosterone affects the Sertoli cells, and this is the ABP binding to the testosterone. Testosterone then has negative feedback. So we have negative feedback of the testosterone. It inhibits LH and FSH, both LH and FSH, but this is fairly weak, this effect. Much stronger is the inhibition of GnRH directly from the hypothalamus. On the other side of the loop, the pituitary is also secreting FSH. And the FSH is stimulating the production of the sperm here, and then be deposited in the seminiferous tubules, and it's affecting all this production of ABP, etc., from the Sertoli cells. So you get the sperm production here, and then also you get the release of inhibin. And inhibin comes back and inhibits FSH release, and this is actually a fairly strong loop where you have much weaker effects on the hypothalamus and GnRH secretion. So those are the endocrine loops that are taking place in the male. Yeah.
So inhibin only affects FSH.
Correct. Yes.
So is estradiol also produced by the Leydig cells? Because
Not much. Very, very little. So it really isn't.
and testosterone and estradiol are both feeding back negative.
Correct. Yeah.
When is inhibin released?
It's-
It knows.
What's really, it's, come back here. Where is it? Here. So, this is where the FSH is stimulating the inhibin from the Sertoli cells, which is released right back out. So it's really part of this negative feedback loop where you get this release, the production of the inhibin that immediately comes back and starts the negative feedback loop.
But it does that year round and not like in a woman when inhibin is only released when she's ovulating.
Correct. Yep. So, the males, male mammals, we don't have to worry about the cycles that are present in the female. Except for, and we'll talk about this a little bit later, except for species that don't breed year round. Okay? So humans are one of the few kinds of species that can breed year round. Yeah.
So is negative feedback acting more strongly on FSH in this case because of inhibin release?
Yeah.
Okay. So that's where, so we still see that like small peak in FSH, or maybe not peak, but like smaller levels of FSH and larger levels of LH and testosterone.
Yep. Yeah.
So for sperm production, you need both LH and FSH because FSH does the mitotic part, and then LH results in testosterone, which results in the meiotic part.
Right.
Okay.
So in terms of sperm production, is LH having a direct, indirect, or permissive effect?
LH? Permissive?
Yep.
Indirect, because it's causing the production of testosterone, which is then
Correct. So it's an indirect effect on sperm production. Yep. Another little fun fact for you. At its peak, the average American younger male produces about 7, I think it's nanograms per deciliter of testosterone per day. And that the testosterone, because of its potential for abuse by weightlifters and etc., is a scheduled drug by the FDA. So you have to have a prescription in order to get testosterone. And you can then be arrested for having testosterone in your possession without a prescription. And the 7 nanograms per mil of testosterone that we produce naturally is above the legal limit defined by the FDA. I find that rather amusing. So we are all legal. All the guys in the room are illegal. Okay. Any other questions? All right. For the last little bit here, I want to ask you a question. I'm going to show you a little data in which we have guinea pigs. And this is sexual behavior. And there's this an experiment that was done in which they had 3 different lines of guinea pigs. Ones that had high, one that had medium, and one that had low levels of sexual behavior. Now, we want to show that this behavior is in fact regulated by testosterone. So what do? Yeah?
Extirpation replacement.
So with the extirpation, how do you extirpate testosterone?
Castrate.
Castrate. So if we castrate these guinea pigs right here, so I'm going to castrate, what do we predict will happen? What's going to happen to the high, to the medium, and the low? Yeah?
They should all drop and pretty much be about the same. Like the line should pretty much like level out.
So they should all drop down to essentially 0? Is that what you're predicting? That they're going to come down like this?
Something like that.
What do people think? That's the hypothesis. Yeah?
I think if we're trying to predict if it's from testosterone is leading to the sexual behavior, that would probably be the case if it's being regulated through that. And you castrate and there's no testosterone being produced. That would be similar to how you would, if you were castrating chickens to make the caprins, because that should be one of those kind of, I guess, bioassays we would say.
Okay. Yeah?
Would the null hypothesis be that there would be no change in sexual behavior?
Correct. So the null hypothesis, right, if this was not regulated by testosterone, then these should stay equivalent. People happy with that? See how that would work? Remove the testosterone. If this was a testosterone regulated behaviors, then we should get basically wiped out. So that's our first half, right? That's our extirpation. Now we're going to do the replacement. How do you want to do the replacement? Yeah?
Do them all the same now.
Okay, let's give them all the same amount. Same amount of T. So what do you expect to see? What's your prediction? They're all going to go up? You expect to see this? Yeah? Yeah.
Should they all go up to their initial levels because like maybe the high strain had like more receptor affinity for testosterone or more sensitivity for testosterone?
Okay, so that's hypothesis one. I'll use a different color here. What happens if we have, instead, we do this? We have our high, middle, and low again. Okay, now I have two hypotheses. Which do you think is more likely? Yeah?
Could it be possible that we started off with the guinea pigs having different levels of testosterone?
Could be. We didn't measure it. But in fact, but we're going to put the same amount back in. So are we going to get the same behavior in all three groups? Or are we going to get three different behaviors? In the high, middle, and the low. Yeah?
Like someone just said, it depends on if there was either, the difference in the stratification between high, medium, and low is due to them having more production of testosterone, like a stratification, or like someone had said, is there actually more receptors for the testosterone in the brain where, the sex drive is? So if it is the latter, then we would see a return to the normal levels. But if it was based on production of testosterone, they would all be the same.
Yeah?
Can you determine that by doing like a third sample with like injections of estradiol and see if that still result in 3 different levels of sexual behavior?
Yeah, we could try doing something like that. This study was actually done in 1952. So we didn't know that testosterone was converted to estradiol yet. But now, if we wanted to repeat this experiment, we could do that. You guys want to see the data? Yeah? Okay. Here are the actual data. So we have here's our high, medium, low, and what we see is they all come down to essentially 0. Notice it takes a while though. This isn't time in weeks. This is, they were castrated at week 10, and we don't get to 0 until week about 21. So it took 10, 11 weeks, almost 3 months before their sexual behavior came down to 0. Then they added the same amount of testosterone to each of these, and what you see is the recovery of that stratification. So in fact, we recovered the high, the medium, and the low strains. Why do we care? What does this tell us about the actions of testosterone? Yeah?
The conversion of testosterone into estradiol is a way to fix that.
Yep, certainly tells us that that's going to be the rate limiting step. But let's back up even higher and think, what does this tell us globally about how testosterone works? Yeah.
Testosterone isn't like a direct, doesn't have like a direct effect.
Well, it probably does have a direct effect, or at least an indirect effect through estradiol. But we didn't know that yet in 1952. But what does this tell us about how testosterone functions? Yeah?
It has like organizational effects that lead to different activational effects.
Okay, exactly. Which ones of these are the organizational effects? These. It tells us organizational. Testosterone has major organizational effects because it is what is creating these high, medium, and low lines. And it also, we show from the castration, has activational effects. This was the first study that first showed that hormones could have both activational and organizational effects. This is an incredibly influential paper that came out in 1952. And talking about the dichotomy between activational and organizational effects. And testosterone is the classic hormone, partially because it was the first one that was shown, but classical has the functions of both activational and organizational effects. Okay? Out of time. So what we'll do is we'll start on Monday, we'll start working on female reproduction.
There's